Welcome to Biocomputation with Max. I wanted to share something more positive about how COVID research can be enhanced. There is some concern about where we are in terms of competing with COVID, and the investment is still often remains to be made. Specifically, microphysiological systems. Why would we go back to an animal model? Some of the publications in premier journals like Lancet now talk about the need to track the variants on multiple vaccines and to fast track identification of a suitable animal model or assay or marker to correlate with these studies. The disease is highly specific, highly humanized, in fact, even personalized. So the suitability of human models, of non-human models, is very limited. So, for example, even non-human primates, while they provide some kind of systemic capability, they're certainly not human. And some clinical trials have shown that with a devastating effect. Human cells in the dish, often on flat, hard plastic, have no systemic relevance. So some providers have been focusing on building tools that would reproduce human physiology on the dish. And so here's a drawing and template from a German company, Tissues, about this universal physiological template connecting multiple organs with fluidics, which is going to reproduce system-wide immunology, for example, or some toxicology. So as I mentioned, COVID is a highly individualized immunopathology. In fact, on our website, you can take a look at an extensive white paper on this subject, as well as a list of biomarkers themselves that over a year ago I thought were top candidates and were needed to be accumulated in integrity, meaning that both cell-based biomarkers, inflammatory, coagulation, metabolic, and others had to be measured. In fact, some of these, like acute phase proteins, C-reactive protein, and procalcitonin, are being used diagnostically for detection of COVID in India, because antibody detection can be so unreliable. And PCR has its limitations and perhaps availability challenges as well. But this is not a very specific detection. It's the same as giving anti-malarial drugs to anybody that presents with a fever. It may be a reasonable practice in some areas of sub-Saharan Africa, but is likely to lead to overtreatment, and certainly in other areas it would be, not even considered. The disease is very complex, as you can see from some of the nature of the biomarkers here, affecting blood coagulation through some extreme inflammatory signaling, recruiting cells in a cytokine storm that elicits this invasive behavior. So this dance is way too intricate to reproduce in other animals. It's not efficacious, it's not humane. But even at the system level, we need to realize that there is a lot of variability between individuals in their immune response, which drives the risk factors and ultimately treatment itself. So these systems that we're talking about for life sciences need to be used not only for development of drugs, but also for personalized medicine. So that's where these applications are, and also that there is economic case to be made for it. So you can read more about the benefits of these models of tissue versus use of animals in our blog. Lastly, I wanted to finish with the slide for actual tools that we provide, and they are very good for pharmaceutical companies and contract research organizations. So here are two examples of microphysiological systems. As you can see, they look very different. Some have a more simplicity and lower price point, perhaps emphasize throughput. Others may emphasize precision, certain ease of use, and have a touch screen and provide visuals. These two pieces of equipment are used for single cell sorting and dispensing. So that could be part of a workflow for a single cell sequencing, for example. So all of these tools are necessary even without COVID for challenging disease models such as neurodegenerative diseases or cancer. But COVID is a very large scale problem, although it behaves as a rare disease in the frequency with which severe cases strike. So another reason for utilizing these systems is that only system-wise analysis is suitable for certain system-wise or system-wide therapies, such as cell therapy or bispecific antibodies. And there are many reasons for that. For example, one of the otherwise very successful cell-based therapies has a significant neurotoxic potential. In fact, their own website talks about all of these highly prevalent conditions. The most common is cytokine release syndrome, which is what happens in COVID. That's what induces the immune system to attack. So there's a commonality of the reaction, if not the initial cause. This one is induced by the treatment, unfortunately, in many cases. And also a substantial fraction of cases show significant high-level neurological events. So this is something that requires further investigation and may in fact be personalized. So that's where these microphysiological systems can come in to help improve otherwise a very poor track record for neurodegenerative diseases and cancer, personalize them and COVID. So there's hope. Stay tuned. Thank you.